Howdy, and welcome to Your Brain, a Personal Tour. I'm Steve Saltwick, and I'll be your tour director. In this six-part seminar, we're going to tour the machinery that is sitting between the ears of every single person listening, your brain. It's both a marvel and a mystery. Now, the marvels we'll get to in a minute, but the mysteries will not only intrigue you, they'll give you some really cool snappy patter you can use at cocktail parties. But before we get to that snappy patter, let's start the seminar with three important questions we'll try to answer. The first question is, why should we study the brain at all? Now, from my perspective, there's many reasons. But the first for this seminar is to better understand the impact of trauma, and especially how to recover from that trauma, if possible. Amazing progress is being made here, and we'll highlight aspects in each of our seminars. The second is to better understand perception. How you perceive an object gives fundamental insights into thought and memory. And then finally, you get that pretty cool cocktail party chatter. And let's talk about that. For example, why does holding a cold drink in your hand affect your judgment of other people? What do nuns do or not do that help prevent Alzheimer's disease? Just what kind of moral decisions does a 6-month-old make, and they do make them? Why is the fact that your brain has single cells that recognize Jennifer Aniston or Ryan Gosling or Homer Simpson a sign of an advanced brain? And finally, why don't lions eat everyone on a safari vehicle? This seminar will provide answers to these questions, and trust me, they're great conversations, at least at some cocktail parties. Now, how can brain science help us understand perception? Well, here are the main points is that perception is prediction. Take a look at this figure. It's called the Penrose triangle. At certain points, your brain will predict that it is a triangle. And it turns out that is when you perceive it. There's many examples of this, and it shows how your brain works. Also, by understanding perspective, it gives us a better handle on explaining the impact of certain strokes and, more important, ways to alleviate the impact of brain damage with technology. Now, the third question is, can the study of animal brains really tell us much about human brains? We will study the brains of many animals in this seminar, especially the rat. Can the rat brain really tell us that much about the human brain? Well, yes, when it comes to the structure of thought. Believe it or not, you think like a rat. Perhaps more in depth, but basically the same process works in your brain like the rat brain. And we're going to study this in detail in Seminar 2. The same is true for memory. We'll study that in Seminar 3, but then, of course, know for human unique aspects of your brain, of which there's only 3 in my opinion, and we're going to study that in detail in Seminar 5. All of these seminars are on YouTube. You can access them with the playlist. So if you want to review seminars, or if we're having an in-person seminar and you missed one, you can look at it again. And I also include a reading list which recommends certain handouts and books if you want to continue the study. Ready to begin our first seminar? Well, let's begin. Our seminar today concerns perception and movement. My one-sentence summary of this seminar is, what you see is not what you get, nor how you act. What our eyes see is not what we perceive. How we think about moving is not how we move. How can that be? Well, we'll find out today. And finally, the inevitable fine print. I am not a doctor. I will answer questions as fully as I can, but do not take my answers as medical truth. Do consult with your doctor as appropriate, and please don't sue me. Now, we begin with a document that's 4,000 years old. This papyrus was written by a doctor in ancient Egypt and records his observations and prognosis on a very wealthy Egyptian. Called the Edward Smith Surgical Papyrus, it contains this hieroglyphic. This hieroglyphic is translated as brain. So this is the first recorded use of the word brain in a written language. So the study of brains have fascinated mankind for 4,000 years or more. Much has been learned, but much remains to be learned. My task in this seminar is to distill as much of this information as I can into 6 seminars of interest. As an example of what we've learned over 4,000 years, allow me to introduce Roger Frisch, a professional violinist. Roger began suffering hand jitters which dramatically affected his playing, potentially ruining his career. He turned to the Mayo Clinic for help. The decision was to apply direct brain stimulation. Now, this involves inserting electrodes into Roger's brain while he's awake in the operating room. Let's take a look.
During the procedure, Dr. Lee inserted electrodes into Roger's brain and moved them to the site where the tremor originated. Once there, electrodes fire electrical signals to hopefully stop the tremor. By measuring Roger's movements, the accelerometer told Dr. Lee if the electrodes were in the right spot. There was no guarantee it would work. Thankfully, it did work.
Now, it turns out Roger can turn the brain stimulation on and off. Let's take a look at that.
You can see the difference. With the click of a controller, Roger can turn the electrodes on and off.
And that's as smooth of a bow as I can play right now.
But when it's on, Roger's mastery of the violin is absolutely clear.
So the impact on Roger, the relief of human suffering afforded by this procedure, is why we study the brain. Now, the violinist needed complex surgery to help him, but recent research noted in the premier journal of medical science, The Lancet, has shown how the study of brain can really pay off. The affliction is depression. I bet you may know someone who has a family member suffering from this terrible condition. A new technique called transcranial magnetic stimulation is bringing new hope to the 30% of patients who are drug resistant. I have a personal friend whose son has this condition, and TMS brought dramatic relief without surgery and at much lower cost. Again, this is why we study the brain. I first became interested in the brain in the early 1970s as part of the biopsychology program at UT Austin. I was privileged to work in the lab of Dr. Michael Gabriel, and we published some of our research in Science. I left UT in the late 1970s and took a tour through the high-tech industry. It was great fun for a while, but even more fun was returning to UT and reestablishing contact with my inner professor and developing this course. If you're wondering where Steve Salwick is in this picture, well, here I am with a little more hair, a lot more attitude, and a little less weight than the one in front of you today. Now, your brain is unique. Here we see a schematic diagram of the nervous system, and one thing about your brain is that it is enclosed in a skull. It sits atop a spinal cord protected by vertebrae. Now, nerves exit the vertebrae and go to muscles as well as other internal organs. This occurs all throughout your body. The organs could be sexual organs, your digestive system, your liver, organs such as the heart, lungs, kidney, your stomach. Other nerves pass information back to the brain from sense organs like your eyes and mouth. If we look at your brain once we remove the skull, this is what we would see. The frontal lobe is at your forehead. The occipital lobe is at the back of your brain. In general, there's not a mapping of function to brain lobe. Yes, visual processing is primarily located in the occipital lobe, and the executive functions tend to be in the frontal lobe. But most everything is spread out all over your brain. That gives your brain a great deal of resilience to withstand damage and trauma. The size of a lobe is not an indicator of brain cell population. The small bit of your brain at the lower right, the cerebellum, contains 50%, half of all your brain cells. The cerebellum controls fine-grained pattern movement, e.g. like a tennis swing. So if you ever want to show someone that you can use 50% of your brain, strum your fingers or swing a tennis racket. That will show them. Another way to look at your brain is to see how parts of it are connected with one another. This is called the connectome. The brain on top seems to be connected in a vertical fashion, and the brain on the bottom seems to be connected in a horizontal fashion. These connections have effects on behavior. The vertically connected brain is typical of a male brain, and the horizontally connected brain is typically of a female brain. These connection geometries probably explain some of the fundamental differences between the genders, including motor and social behavior. Now, if we were to go inside the brain, we will find cells that make up this marvelous organ. We would see billions and billions of specialized cells called neurons. These neurons are electrically excitable and transmit impulses of electricity down long, skinny parts to other neurons. When that electrical pulse, the little blue pulse, travels down to the end of the snaky bit, it causes a chemical to be released onto the next neuron. That chemical may or may not cause the next neuron to get excited. So neurons are both electrically excitable as well as chemically excitable. There's two ways we can control a neuron, and we'll use that fact in experiments when we study later on. This also means that neurons can become dysfunctional in two ways, electrically and chemically. Since neurons use electrical signals to communicate, we can actually record this in living brains and amplify the sound. I'd like to play you something that we might hear in a laboratory if we were recording from brains of a living animal. There's many different rhythms. Some are quite fast, some are quite slow. Some are silent and burst. Each style has a place in the way your brain operates. Since it's fully enclosed in skull, your brain needs some help to interact with the physical world. That comes from the peripheral nervous system, a system of nerves that's outside the skull. Everything your brain senses is carried by incoming sensory nerves called afferent nerves. They carry familiar senses like sight, hearing, and taste. But note that internal sensations from muscles and organs are carried to the brain as well. These sensations can help explain some surprising experiments, which we'll see on the next slide. Behavior is caused by efferent neurons leading away from the brain. The vast majority of our time in this seminar will be spent on behavior called by muscles. We'll not have time to give justice to the autonomic nervous system, the fight or flight system that you probably studied in high school biology, but I do want to highlight how the feedback loop of these systems are incorporated into higher order behaviors of learning, emotion, and memory. Now, a strong case can be made that feedback from your internal organs and senses underlie emotional responses and memory. Stated another way, your thoughts are not just some abstract symbols and language, but incorporate internal bodily sensations. Some examples might help. If I ask you to evaluate the personality of a stranger while you have a cold drink in your hand, you will tend to find the person colder than if your hand is empty. And yes, if you have a hot drink in your hand, you'll judge them to be warmer. If you evaluate a job candidate with her resume and a heavy clipboard, you will judge her more serious than if the resume is a piece of paper. Our inner gut feelings are an essential part of our brain's experience, and that fact will be important as we explain and explore how our brain interacts with the world. We can also use this connection to reduce implicit bias, a very positive action. Your brain evolved. What we show on the left is some representative brains across a wide variety of species. The human brain is at the top left. A mouse brain is at the bottom right. For those of you that are dog and cat lovers, look at the middle row. The dog people can be amused that the dog brain is slightly bigger than the cat brain. I know that dog people will say that's obvious from the start, and the cat people will say that's a small cat. I'll leave it to the dog and cat people to fight that out. While size does matter, it really doesn't relate to intelligence. There's some very smart birds whose brains would be down at the bottom right. What matters is the functional areas of the brain, and there's a complication on that point. Brains differ in their organization of functional areas over the course of evolution. To use an example, suppose our common ancestors had a red, blue, yellow, and orange set of functional areas. The family tree on the right shows how those areas might move around in terms of size and placement. The key aspect of this diagram is the appearance of new areas. They bring new function. These new areas are essential underpinnings of the different intellectual capabilities. So what you're seeing from this image is from the Glass Brain Project. This is an actual real-time image of electrical impulses in the human brain that's being studied in a laboratory. What is it doing? How does it sense and perceive the world around it? Is what it sees, or in other words, what it senses, what you get or consciously perceive? Or is it something different? To answer these questions gives us our first main insight into the way the brain works. Your brain senses the outside world through a series of awesome sense modalities. We learned about several of them in high school, and they're familiar. Sight from the eyes, hearing from the ears, etc. Most come to the brain through a relay station called the thalamus, number 2 on the diagram. Smell comes direct. That means you can smell when you sleep. We will use that fact about smell in some experiments later. I want to draw your attention to the somatic senses. They're not as familiar to many of us. The key somatic sense I want to talk about is called proprioception. This is a sense of what the muscles of the body are doing. The top left-hand part of this slide shows a silhouette of a bicep muscles of the arm. A portion of that muscle is then expanded. The muscle is innervated with some sensors called muscle spindles. Nerves leading out of those spindles give your brain a sense of the state of that muscle. Spindles are not the nerves that make the muscles contract. They're different and they're not shown. Muscle spindles give your brain, a sense of muscle tension. It turns out to be extremely important not only for your movement, but also for your perception of self. Your brain tells your body to move and then gets feedback of the state of movement from the spindles. The brain's interpretation of that feedback is believed to be extremely important in creating a sense of self that we typically assign to consciousness. I find it very, very interesting that such low-level sense within your muscles might be so important as to be the underpinnings of conscious perception of self. The somatic sense of proprioception can be used for dramatic effect in areas that might seem surprising. Let's look at one example. Here we'll use the feedback path from facial muscles to reduce implicit bias. And this is implicit racial bias. How is that possible? First, it's important to look at how we measure bias. Something called an implicit association test is typically used in this area. In this test, the subject presses one of 2 keys to categorize either words or picture of faces. The words must be sorted into good or bad categories, and the faces must be sorted on the basis of race. In one condition, the same response key is used for both good words and white faces. In a different condition, the same key is used for good words and black faces. The key measurement is the difference in response time between the 2 conditions. Typically, subjects respond faster to pairings of good with pictures of faces which are the same race as the subject. Therefore, would be measured as showing its implicit bias against the other race. It's important to note that the implicit bias test is more like measuring your blood pressure than measuring your height. All sorts of variables can affect the results, but the test is generally accepted as a valid member of implicit bias. So, on with the test. Could manipulating the somatic sense actually affect bias? Could it reduce bias? That's the $64,000 question. And to make a long story short, it could. What was the manipulation? Having the subject hold a pencil in their teeth so that they smiled. The act of smiling reduces implicit racial bias. Isn't that cool? So unleash your inner optimist and smile. It will do the world and you good. But humans are really visual animals. Hearing might be the most regal of senses, but the sense we typically are most aware of and use is vision. The nerve pathways from the eyes lead to the back of the brain, that's the occipital lobe, very directly. They crisscross and make one synapse or connection on the way. Most visual processing begins with the back part of the brain. The central part of the eye is very dense with sensors, so the central part of your vision field gets the most resolution, and that's where you see most clearly. But there's an early warning system in the visual path. Just after the nerves exit the eye, a special path connects to a threat detection network in your brain. The resolution of this path is not very accurate. It goes on general shape. For example, snakes, probably the first predator of mammals, and fearful eyes as detected by large relative areas of white. These early warning signals are detected by the path, and it's very helpful in reacting to a potential saber-toothed tiger that you might have encountered in the past. And it's also very involved in implicit bias in the present. More on that a bit later. Using computers, we can map where the central part of our eye, called the fovea, is focused. In this diagram, the little red dot basically tracks where the fovea is focused. That red dot is where the brain is getting the majority of information about the visual field. Let's play a short video and watch where the fovea tracks. So what did the fovea focus on? To me, it was almost exclusively the faces of the two people. You didn't see the red dot survey the room. You didn't see the red dot look at the clothes. It almost exclusively focused on the faces. Other parts of your eye did take in the visual scene, but with far less detail. Your brain builds up a scene over time, and some of the parts of the scene get virtually all the visual attention compared to other parts of the scene. That's an interesting way to build a picture of the world. After the eye has processed the visual scene and nerve impulses have gone to the back of your brain, how does the first stage of visual processing occur in the brain? Well, the first level of visual processing is the detection of elementary objects in very precise locations at very precise orientations. Here we see a monkey in an apparatus where we can study his visual cortex in the back of his brain. In front of the monkey is a screen. We can project lighted bars or other objects on the screen, and we study the response of neurons when we present those visual stimuli. On the right is a figure of what a representative brain cell or neuron does in response to a particular bar of light. This one is preferentially tuned to a particular stimulus, a rectangle oriented at about 40 to 45 degrees from the vertical. When the bar of light passes this visual field of the cell, it responds with a burst of activity, like this. That's a very robust sound. If that stimulus is vertical, you only get slightly less response. Tilt it the other way, and you get no response at all. This is an excellent description of how first-order visual processing occurs. Neurons in your visual cortex are tuned to very precise stimuli at very precise places at very precise orientations. What happens to the brain process after this first level of detail? Well, remember, I said perception is prediction. And that's what we see even at the first level of visual processing. Look at this lattice work. Focus on one section. You will see a black dot at the intersection of the lines, typically only one, maybe two dots at a time. Move your eye around the figure and you will continue to see only one or two dots, but they're everywhere at the lattices. Why? Because your brain is predicting what is happening in the pattern. It's filling in blanks because it is assuming that the pattern exists. Now, various other areas at the back of your brain, the primary visual cortex, analyze the visual field for further basic information. Color. What are the areas of color? Motion. What parts of the visual field are moving, and if so, how are they moving in relation to one another? detecting that those spots moving behind the little bush next to you could be a saber-toothed tiger, well, that's a good thing for your brain to be aware of. The form of the image is analyzed. Does it correspond to objects we might recognize from memory? Finally, you have two eyes, or a binocular species, and we can detect depth of objects, so a 3D image of the object is built up in parts of your brain that give us a sense of solid form. Now, what happens next? The next stage is breaking down the visual image into fundamental building blocks. Examples of these building blocks are given on the left-hand part of the slide. They're called geons. They're canonical objects. You can think of them as Lego blocks of fundamental shapes. By taking these fundamental shapes and merging them together, the brain is able to build representations of more complex objects. For example, if your brain wants to build an object of a paint or water bucket, it takes the shape of a curved handle, number 5 here, and a cylinder, number 3 here, and puts them together to form an object of bucket. There's good evidence that the fundamental analysis by your visual cortex merges together geons in various other ways to form object representations. This is a core principle of your brain. Objects are analyzed all at the same time into simple canonical forms, and then these canonical forms are used to build more and more complex objects by additional layers of your brain. Now again, if perception is prediction, then these building blocks are used by your brain to build an internal model of an object. This is illustrated by the following video. What we will see is the construction of a line object in two-dimensional space, and then we're going to alter it, all right, and then take a look at it from various perspectives. Note how your brain builds a three-dimensional object from elements on this simple piece of paper. Even though you saw this two-dimensional set of lines being constructed, your brain builds an internal model that this is a three-dimensional object, and that is the object that you perceive. Now, objects can be pretty complex. For example, one object reported in the scientific literature is the Jennifer Aniston neuron. This particular cell responded to pictures of Jennifer Aniston, not just full frontal face, but also side views like you might get from a police lineup, line drawings of Jennifer Aniston, even the name Jennifer Aniston written in block letters. Now, not to be sexist, similar responses have been made for patients viewing pictures of Ryan Gosling, Tom Cruise, or even Homer Simpson. Now, you are not born with neurons for Jennifer, Ryan, Tom, or Homer. Your brain learns to group objects as a function of experience. We can understand this by studying the perceptions of newborns. At 6 weeks of age, newborns have no conception of angles. They react to the orientations of lines. So these two figures are equated since this line and this line are the same. However, by 14 weeks of age, babies have grouped line segments into objects where the interior angle is significant. At this age, these two figures are judged the same since this angle and this angle are the same. Now take a look at this diagram. What do you see? There's two ways to see this. One way is a set of blue lines and a Pac-Man-like circle sitting on a flat plane for no apparent good reason. The other way to look at this object is a solid white triangle suspended above a blue triangle and three circles. And that's the way most humans would describe what this is. It's not an arbitrary collection of line segments and funny-looking circles, but familiar objects in space. What you've just done, is a conscious perceptual decision. Your brain decided that spatial arrangement of objects, it analyzed the geons present, and it basically decided that they represented forms of triangles and circles arranged in space, just as we talked about it in the preceding slides. It analyzed for color, it analyzed for form, it analyzed for depth. The reason this figure is important is that in most research, it now suggests when your brain makes that kind of perceptual decision, that is when these objects really enter into consciousness. we're really not aware of how the brain is doing these fundamental analyses, the line segments, the geons, what have you. That's not conscious in our experience. But when we decide on a perceptual framework, that's when we build the geons into objects positioned in space. And that's when we enter those objects into consciousness. Recall that we're primarily visual beings, and that visual input to our brains have a significant effect on how we predict perception. One dramatic way to demonstrate this is called the rubber hand illusion. Let's take a look.
Oh, I have an experiment that we're going to do today. Is I'm going to train your brain to believe that this hand, this arm, is your arm. You see we've got this divider here? So right now you can kind of see your arm. You see the arm there? And I'm going to cover over part of your arm. Now I'm doing that because your eyes talk to your brain. And your eyes tell you this is what's happening. So now, can you physically see your arm, your right arm here?
No.
Great. So I'm going to take these two classroom rulers. So we're going to train the sensations. You feel the rulers touching your hands because it's now becoming one with your mind. That this right hand is your right hand, right? You are believing that as I drag the ruler on your right hand there,
It totally
that it is
feels.
in fact in your brain your right hand.
It totally feels like my hand. That's crazy.
Feel the sensations line up with each finger.
What? Whoa, yo, this is crazy.
Yeah. So now if I just do like a little tap, right?
Yeah.
Feel the knuckles. Feel each digit.
Oh, that's
Okay, this is where.
crazy.
Yeah. Now I'm gonna do this one more time to train your brain.
Okay.
Right. So you feel your thumb
Oh, okay, yeah.
and your index finger. Middle finger.
This is nuts.
Your ring finger.
Oh my goodness.
And your pinky finger. And now you're gonna feel it when I tap it. You feel that?
Yeah.
And that.
Whoa.
I'm gonna let you in on a little secret.
Yeah.
I have not been using the other ruler.
What?
Feel that?
Yeah.
I am not using the ruler.
Whoa. What do you... I mean, it's crazy. It's not... Oh! Whoa,
You
and
felt
even
that,
like...
right?
Oh, no.
Now, check this
Oh.
out. We're gonna do a little test. Now, again, this is all in your mind. I'm retraining your brain to feel sensation.
Okay.
But your brain is thinking that it's feeling it.
Okay.
So even if it gets a little, what would be considered painful, I will not be harming you because of course I'm not going to be actually hitting your right arm. But your brain really believes that this is in fact your right hand.
Oh,
right?
this is making me nervous.
All right, here
Okay.
we go. I'll start lightly. Feel that on the thumb.
Whoa, yeah. Oh. Whoa.
Ring finger.
I'm totally feeling it in this hand. It feels...
Pinky.
It just... Oh. Wow, this is so
Back
strange.
of the hand. You feel that?
Yeah. I want to move my hand, but it feels like I can't. What's that for?
Now again, I would never on purpose hurt you, right?
Okay.
And I would never take a hammer and bash your fingers to where they would hurt, right?
I'd hope not.
But again, your brain is going to tell you that you're going to feel this when in fact I'm not actually going to be causing any damage to your hand.
Okay.
Right?
Okay.
And we'll start lightly again. Okay?
Okay.
You ready?
Yeah.
Here we go. Very lightly. Here we go. There.
Oh, it's so weird. Oh,
Oof.
oh.
Oh, the infamous thumb when you're hammering. Oof. Missed the nail.
Oh, you know, it feels like an electric shock. It's so strange.
Doesn't it?
Yeah. Oh, even when I touch myself.
Yeah.
Oh.
Yeah. Watch this. Watch this. One last electric. Watch this. You ready for this one?
Okay.
Guess which finger I'm gonna hit.
Okay.
Guess which finger. See if you can tell me which one it is.
Middle.
Ooh. Now again, I did not actually hurt you, did you? You didn't feel
No.
actual pain in your real hand. But for an instant, your brain told you that you were thinking that you felt a hammer hitting your hand.
My brain thought that this was my hand.
I'd like to summarize this section of the seminar using a well-known celebrity, Homer Simpson. We'll use him throughout the seminar to help reinforce the main points of each section. So here we have an x-ray of Homer Simpson. You can see his brain is slightly smaller than the brains we've been discussing, but that's because his brain can only hold one or two important points at any one time. So Homer's summary statement is that your brain senses stuff, the line segments, geons, etc., but you perceive objects which your brain predicts. This is a fundamental property of sensory perception. What you see is not necessarily what you get, because you only get objects. Objects enter consciousness, and that's when you're really aware of things, when the various sensory modalities we've been analyzing to the level of an object, and the brain predicts the perceptual hold. An object in space and time. That's the main message from this portion of the seminar. Now let's turn our attention to the other end of the spectrum, movement. We interact with the world around us by moving muscles. We change the position of our limbs. We turn our head. We focus our eyes. So in the last part of the seminar, we're going to look at how does the brain do that? And more importantly, we're going to see if there's something that is equivalent to objects as we discovered on the sensory side of the equation. We saw how initial sensory input was analyzed at a very low level and then abstracted into objects. In fact, most objects were extracted into objects of objects. Well, is there something that goes on in the movement system like that? Let's find out. Our first hint that there might be something like movement objects was discovered in an experiment from 1977. An enterprise young psychologist asked several subjects to write the same sentence with a variety of appendages. This particular sentence is a palindrome, so it says the same thing whether you spell it from left to right or right to left. It's clear to me from looking at this picture that there seems to be some kind of template that represents cursive writing. The template is expressed in a variety of muscle movements. The right hand with restrained wrist, the left hand, the teeth, and even the right foot. Now clearly, fine-grained muscle movements do vary across these samples, but I would look at this and say there's some very interesting evidence that points to a particular movement template. That template is then expressed by different movements of different muscles. But again, the movements are expressing the same object, the sentence. In the search for movement templates, there's always a controversy. Does a purported template reflect the actual goal of the movement itself, or is it really just the muscle commands in some fashion? We need to find neurons in the brain that reflect the template, not the muscles. So let's look for that next. The classic experiment that showed, yes indeed, there are movement objects used a very clever idea. The idea was to use two different kinds of pliers. Normal pliers are shown at the top half of the screen. To pick something up or grab a piece of food with it, you would squeeze the pliers. The bottom of the figure shows reverse pliers. These functions exactly the opposite to normal pliers. To open the tongs, you've got to squeeze them, and then to grab the object, you relax your hand. The set of muscle movements between normal and reverse pliers are exactly the opposite to accomplish the same goal. If we could observe a neuron that represented grabbing food or an object, and it responded the same way using both the normal and reverse pliers, we would have definitive evidence of a template for movement. In other words, a movement object. We see here a monkey using such pliers to obtain bits of food. The top video shows the monkey using normal pliers. The red lines coming out of his eyes are a computer animation of where the monkey is looking. This is not Super Monkey. They're not lasers coming out of his eyes. This is just where he's looking. He's grabbing the food and bringing it to his mouth. Remember, he is squeezing in order to grab the food. Let's take a look at the same monkey using reverse set of pliers. In this instance, you can see he's relaxing his hand to actually grab the food. In the same monkey, we recorded cells that correspond to grabbing of the food. The red arrow shows that in time when the actual grab took place. Neuronal activity is shown in two different fashions. On the top of each graph, there's a little tick mark every time the neuron is active. There are many trials, so each time the monkey reaches for the food, there's a different row of tick marks. Below the tick marks, there's a histogram of activity over all the trials. Now let's look at Unit 210. It's very active just as the monkey grabs the food with the normal pliers. Likewise, a different neuron in the same monkey is very active as the monkey grabs the food, again with the normal pliers. Now those same units were observed at the same point in the movement when the monkey was using reverse pliers. Remember, the muscle movements are exactly the opposite. However, the behavior of the units in the brain are exactly the same. Several other neurons were found that had similar activity. Basically, these neurons code for get the food. They encode for the act of grabbing that piece of food regardless of what muscle movements are involved, and that is an object for movement. Now, many complex movement objects have been found. They're specific areas where stimulation of the brain gives rise to very specific behaviors. These are movement objects. They're composed of other objects which ultimately result in the movement of a different set of muscles. There's reaching, climbing, leaping, chewing, defensive postures, moving your hand to your mouth. There's a rich repertoire of behavior here represented as complex movement objects. How did this happen? Well, it's a complex story. Not only are there regions in your cortex, the highest parts of your brain involved, but also non-motor areas involving emotional systems, the cerebellum, systems in your spinal cord, and then finally motor neurons that directly control the contraction of the muscles. Many people will spend their whole lives studying this system, but I have an analogy that will convey to you a way of thinking about this whole thing works. Think about a piano. When you push the key of a piano, there's an amazingly complex set of element movements, that transform the movement of that key to the sound that you hear. The physics of how that happens is amazingly complex and very subtle. But understanding the fact that you push a key and out comes a sound gives a good analogy of how a movement object or a vocabulary of movement would relate. It's the fact that there's a high-level concept like a key, and then ultimately a set of movements which produce a vibration of a string, right, that produces the sound. This, I think, is a good analogy for us to consider. Now, recent research has shown that movement objects are constructed by your brain from a grammar of behavior. Syllables of that behavior are shown in the diagram. Research like this will help us better understand the impacts of stroke on movements, as well as the potential use of technology to recover function whenever possible. Now, an example of using technology like this can be seen by studying the movements of this walking rat. Here, significant progress has been made by the design of an exoskeleton to help paralyzed people walk independently. This is one of their top desires. Better understanding of the behavioral grammar will help us improve the mobility of such people over time. A final item I want to talk about is the difference between moving your muscles and imagining moving your muscles. If you look at the top part of this diagram, it shows the areas of your brain that are active when I ask you to strum your fingers. We would basically see two areas of your brain active. when I ask you to imagine moving your fingers, and you don't move your fingers, you just imagine strumming your fingers, we get the bottom picture of brain activity. Notice one area of the brain that is active when you're really moving your fingers physically is also active when you're imagining moving your fingers. This has significant implications for memory. When you remember moving, you activate some of the same areas of your brain that drive the actual physical movement. Memory activates some of the same areas as the act itself. We can use our Homer analogy to explain this in a simple statement. You plan with movement objects and you move with muscle stuff. Almost the exact mirror image of our sensory analysis. We said that you sense stuff and perceive objects that you predict. Your brain's sensory systems go from low-level analysis to high-order objects. The movement system is the mirror opposite. Your brain is planning acts based on high-level movement objects, and they get translated by this complex piano machinery into muscle stuff. Some muscles contract, some muscles expand. Here's the feedback. There's pattern generators for complex behaviors like locomotion with a syntax of behavior. The essential fact here is that your brain is planning behavior with movement objects that then gets translated into muscle stuff depending upon your position in space. Isn't that cool? I'll close with the following observation. Sometimes the motor part of your brain is smarter than the rest of your brain. Look at this screen. I suspect everybody watching would say that the orange dot in the middle of the large circles is smaller than the orange dot that's in the middle of the smaller circles. This is a classic illusion called the Ebbinghaus illusion. If you took out a ruler and measured the size of those orange discs, they would be exactly the same size. But they look different. However, if I built these discs out of wooden blocks and put them in front of you and asked you to reach out with your thumb and forefinger and pick up the orange ones, the distance between your thumb and forefinger as you reach for these objects would be exactly the same. So the motor planning part of your brain knows the state of the real world. The visual analysis part of your brain doesn't know the real world. And that sets up an interesting dynamic. If different parts of your brain can have different opinions about the state of the real world, how does your brain decide to act? Well, that's the topic of the next seminar. So this completes our first seminar. The topic we'll talk about the next is illustrated by this quote from Amelia Earhart, a very famous American pilot. She said, "The most difficult thing is the decision to act. The rest is merely tenacity." We saw today how the brain can have different opinions. Is the size of the orange disc the same or different? How does the brain come to a decision about how to act on that? Today, we studied the movement system, but the decision to act had already been made. Our next seminar is going to be how the decision-making processes of your brain work as it decides to act. I hope you've enjoyed the seminar. See you next time. This is Steve Saltwick.